Hey everybody, this is Mr. N again. I want to do some review on previous worksheet. Uh, the worksheet was about transcription practice, so I'm going to read the question. The last question basically asking about, you can pause the video and go to your homework or worksheet. I think it was about transcription practice. Go and find it and prepare yourself. You may do it yourself. You previously did it. You want to make sure that you understood the concept, and if you did any mistake, I'm gonna tackle the problem. I'm gonna show you the correct answer. So the question says, imagine that the non-template sequence in previous question was transcribed instead of the template sequence. Draw the mRNA sequence and translate it using the codon amino acid table. How well will your protein be functioning? So basically, this time, instead of using the template, you're going to use the non-template DNA, the complementary one. So first, let's see what is the template DNA. The template is here, 3' to 5', TTCAGTCGT to 5'. So now I want to make the non-template strand, and then I transcribe that as template this time. So I want to make the non-template one. I already did something on the top, so I'm going to copy it and you will see it better. Okay, so 5' this time to 3'. It's going to be AA G T C A G C A, and this is the 3'. Remember, I just made the non-template DNA, not RNA. That's why I have T, not U. Let's make it clear. So I just made the non-template, or we can call it cDNA. Mostly when we say cDNA is the one that we synthetically make it. So this is a non-template, but here we want to use it as template this time, based on the question. Alright, so I want to make mRNA now. This is not an mRNA, this is a non-template DNA, which question asks us to do. Camera is here, I made mistake to look at that record button. So okay, so alright, I'm going to do it now. This is the DNA. Again, mRNA is gonna be, use different color. 3'. It's gonna be U C A G U C G U. I went a little bit crooked. So the green one is the mRNA I made from the non-template DNA. Alright, so this time we're considering the non-template basically is our new template. So I made the mRNA. And remember, if my mRNA is directed 3' to 5', when I'm translating it, I should have it from 5' to 3', because translation system reads the mRNA from 5' to 3'. So I'm gonna flip it. If you did from your, I don't know, you did it yourself, you already turned this from 5' to 3', you do not need to flip it. But I'm gonna flip it now. So basically, I put this one U at the beginning. It's just a flipping, just so you should have conformational arrangement in your head. I mean, it's very easy. U G C. I'm more comfortable doing it here. I will show you. U G C U G A C U U. Okay, and here is this is the direction that the translation is going to happen. All right, UGC, UGA, and CUU. Based on the table, I don't memorize anything. The only thing I memorize is the start. Let's hit the camera again. Start codon is AUG, right? AUG is the start codon for methionine. So basically AUG, whenever the translation system sees AUG, amino acid will be methionine, but also AUG is the codon that starts the translation. So this is based on the table I already found it. This is going to turn into cysteine abbreviation. UGA is stop codon. UGA, UAG, UAA, three of them. There are stop codons. So stop, and CUU is leucine. Okay, but there's a trick. Whenever the translation system, the ribosome, whatever proteins are involved, the rRNAs are involved, see the stop codon translation does not happen. So what you get here is just one amino acid, which is non-functional. It's basically useless. All right. So you can say this: the protein, amino acid is made. It's not going to be useful. You're not going to make the polypeptide. Polypeptide is not made. The chain is truncated already. So that's why the final part, I don't remember what was the final part question, but we can see that the final, how well will your protein be functioning? It's not going to be functioning. All right. But if you have used the template, that will be a different story. You may get the chain of amino acids. So it's very important which strand is used for transcription, which gene is used, just like a strand, we have different genes, which strand is going to be used for translating the codons. So for translation, we know that there is a mediating complex molecule, tRNA transfer molecule, that grabs the amino acid on one side and matches with the codon on the other side. So basically, if we have just like tRNA here, the strand itself is here, and basically based on the conformation and the base pairings, because it's single stranded. For example, if you have A here and you have U here, this is gonna just like wrap around each other and gets base paired, hydrogen bonding will happen, and they will go just like 3D shape. Well, the flattened theoretical shape is like this, but the real shape in the physiological in cells system will be like L-shaped. So you can see it has 3' here, which ACC, I'm not sure you can see the ACC. ACC is here. Amino acid will be attached to here and makes the bubble. You don't need to memorize everything here. And the anticodon or anticodon, which will be matched on the mRNA side, is here, 3 nucleotides. Let me show you just like a fork. If you imagine, if we have so if this is the mRNA, and here let's say we have AUG, it's from 5' to 3', 5' mRNA, 5' to, and AUG, remember, is the start codon, right? So the tRNA can see, and the ACC, the 3' side, can be attached to the amino acid, equivalent amino acid. This is methionine. Of course, it should be 3 teeth, but the fork has 4. That doesn't matter for our, depiction. So it's gonna be like this. I hope you see it better. It's gonna be matched like this. So the bottom part is anticodon. The top part on the 3' end, not 5' end, will be attached to amino acid. This is the way it works. All right. You have any question? I can't hear you. All right. So we talked about the basics. Here we'll be encountered with the start of the translation. So you know the ribosome has two parts, two subunits, the small subunit and large subunit. And each of them are composed of rRNA, and rRNAs, different types of 3 and 4. That depends on the eukaryotic and prokaryotic. Again, you do not need to memorize the details. You know where are they made? In nucleolus. Nucleolus is the place that rRNAs are made, and there are a lot because you have a lot of ribosome in the cell. It's the most abundant type of RNA or rRNA. So yes, RNA made in the nucleus. Protein comes from the cytoplasm. It's going to go inside the nucleolus, and the subunits will be assembled and then moves out of the nucleus from via the nuclear pore and nuclear pores and what else? Yes, the mRNA will be found by small subunit first. So they're not assembled yet. Each subunits are separate. So a small subunit will find the mRNA, moves from 5' downstream to 3'. Okay, so the first and just like first docks into the special site near the start codon, and whenever it finds the start codon, the as I showed you, the amino acid and tRNA complex, which we call amino acid tRNA, which are mashed together, combined together by the help of the amino acid transferase. So this charged tRNA, when we have the amino acid attached to the tRNA, we call it charged tRNA. The start one gets inside, matches to the start codon, and it's the beginning of the translation. And then it causes the large subunit to be attached to the initiation part. So here large subunits come to the play. The start one is in the P place. It has E, P, and A. For simplicity, imagine exit. E is exit, that the tRNA will be living without amino acid. Amino acid will be just like get together in the P side, just like polymerization. And A is the site that the new tRNA and the new amino acid will be there. So first, when the large subunit comes, the start codon will be tRNA with the methionine will be in the place, in the P place, and the new amino acid will come here. The new amino acid, by the help of the rRNA, will be matched to the methionine, and the just like when this is happening, a ribosome will move toward the 3'. So 5', 3' moves toward the 3', causes the shift, causes the A tRNA, which was in the A, moves to the P, and the P, which is empty now because its amino acid transferred to the one at the beginning, and the empty one will leave to the, sorry, I'm seeing the camera in the wrong direction. So the one here will go to the E, the exit one, and the empty tRNA will move and leave the complex. So we'll add up one by one and make the chain, polypeptide chain. Here's the general information about that. So rRNA is the one that connects the amino acid together. Regarding, somebody asked me, emailed me about the exam, in college board exam. So basically you're gonna have the 2 FRQ. Don't ask me about the multiple choice question. They have decided not to give you multiple choice questions. So 2 FRQ questions, each of them, the first one is, I believe, 25 minutes, and you have 5 minutes to upload your answer. And when you submit it, I think you cannot get back and edit it. And as you go for the second FRQ, and you have 5 minutes for uploading again. What else I can say is FRQ, no multiple choice. And yeah, basically you saw the FRQs could be just like a scenario based or application based, and they could ask you like A, B, C. That's why it may take 25 minutes. All right.